Thank you very much, Mala, for telling us how we should be going in the future. Now we'll talk to you about what we've done. It's a pleasure to introduce the ICEMRs to you, and this morning's talk will start with Dr. William Moss, representing the Southern Africa ICEMR in Zimbabwe, Zambia, and Zimbabwe and Zambia.
Great, thank you very much.
We'll take 30 minutes.
Thank you very much, and welcome to all of you. I want to give a special welcome to all of our ICEMR colleagues from around the world and around the country for coming and celebrating World Malaria Day with us. I'm going to try to tell you very briefly 3 stories around successes and challenges in malaria control in the Southern Africa region where we've been working. It's very challenging to try to summarize 6 years of work across 3 countries, 4 sites in about 25 minutes. So I'm going to move rather quickly to give you a feel for the types of questions we've been trying to address and the approaches we've been taking. And in line with what Mala said, we've very much tried to integrate across disciplines, across epidemiology and vector biology and parasite genomics to address some key questions for malaria control in this area. So the Southern Africa ICEMR, the title of our program is Malaria Transmission and the Impact of Control Efforts in Southern Africa, and we consist of 8 institutions. This is really a large collaborative project. This is a map that we created for the ICEMR supplement in the American Journal of Tropical Medicine and Hygiene. Tim Shields created this map for us. It shows the location of the different ICEMRs around the world. Oh yeah, I can't use I'll use the mouse, sorry. And we're the Southern Africa ICEMR here. I'll show a close-up of this, and we have our sister ICEMR in Malawi. So the Southern Africa ICEMR consists of different transmission settings, and almost in the same framework that Mala laid out. We work in, maybe I'll start here, in Choma District. This is a very low transmission setting in southern Zambia, moving toward elimination. In northern Zambia, in Nchelange, very high transmission area where the standard mix or package of interventions have been ineffective. We've more recently started some work on the DRC border in Kilwa and Kashobe. I'll mention that briefly. And then kind of in between is this setting in eastern Zimbabwe where there's been seasonal resurgent malaria and some recent success in malaria control. So this is a very critical geographic expanse from extending from eastern Zimbabwe up into the Democratic Republic of Congo. Malaria control and elimination in this setting is going to be critical to achieving global malaria control and elimination goals, and in shrinking the malaria map up from southern Africa into Central Africa in the very challenging setting in the Democratic Republic of Congo. Very briefly, I'm going to describe our methods. We use satellite imagery to establish a sampling frame. This is a satellite image of Nchelange District in northern Zambia. This is Lake Mweru, and Democratic Republic of Congo is on the other side. And we use this to establish a sampling frame, but also to get ecological factors, and we use remote sensing to generate risk maps. I'll show you that. We use both active and passive case detection. Our active case detection is which we select randomly selected households, go to those households and conduct surveys, collect blood samples, and do our malaria epidemiology and entomology. So our entomology is linked with our epidemiology. We also have a passive case detection that Clive Schiff actually started in southern Zambia, where we get weekly SMS text messages from the health centers regarding number of confirmed and suspected malaria cases. So this will serve as the source of data for the studies that I'll tell you about. So I'm going to start in Choma District. This is in the area with very low transmission, moving toward elimination. And this is where our research questions revolve around achieving, sustaining, and documenting malaria elimination. These are hospital data from Phil Choma, who's our main collaborator there, showing pediatric hospitalizations for malaria over a 16-year period almost. You can see a dramatic decline in pediatric hospitalizations. When the Johns Hopkins Malaria Research Institute started back in 2002, 2003, malaria was an enormous public health problem, and there's been this dramatic decline in transmission. And we could probably have a whole session just talking about what drove that decline, but I do want to highlight changes in weather patterns and climate that are really critical for understanding patterns of transmission in this area in southern Africa. Many of you may have seen the recent news on the drought in this area. That's obviously has a big impact on agriculture, but on malaria transmission as well. And what we're particularly interested in now is trying to understand what drives this low-level transmission here, what's driving these cases. And there are two broad hypotheses. One is that these are imported cases of malaria coming from outside the region, or that these arise from some asymptomatic, chronically infected reservoir that kind of feed local transmission. And so we've been working to try to understand those drivers. This documents the decline in parasite prevalence that we've observed through active case detection. So this is going out into the community, measuring the prevalence of malaria by rapid diagnostic test and PCR. And we can also see through this very different data source, this dramatic decline in malaria transmission. And again, we're trying to understand, what's driving this low-level transmission, parasite prevalence of 1% to 2%, largely in asymptomatics through this active case detection. So I'm going to frame this in achieving, sustaining, and documenting elimination. I obviously can't present all the data that we have, but I just want to highlight a couple of the challenges in achieving elimination and some of the strategies that have been implemented by the National Malaria Control Center in Zambia. First, and this is, perhaps comes as no surprise, but for active case detection, HRP2 rapid diagnostic tests are insufficiently sensitive to detect infected individual in the communities. These individuals have low-level parasitemia. We've looked for the HRP2 and 3 deletions in this community and have not found it. We think the false negative RDTs are due to very low levels of parasitemia. We found particularly low sensitivity of the RDTs in this setting of about 20%. Now, to be fair, RDTs were not developed for this use, for active case detection in low transmission settings. They were developed, obviously, for identifying and confirming symptomatic disease at healthcare facilities. But they're being used in this setting for detecting asymptomatic individuals. Now, the National Malaria Control Program in Zambia in Southern Province, where we're working, implemented a reactive test and treat strategy, in part to enhance surveillance and in part to try to minimize this asymptomatic reservoir. So when an index case presents to a health center, the community health worker goes to that index case household and tests and treat with an HRP2 RDT everyone who resides within 140 meters of that index case house. So there's a radius around which this reactive test and treat works, and then they treat everyone who's positive. For the past year, so from January to December 2015, our field team has been accompanying the community health workers and collecting dry blood spots for PCR and extended the radius out to 250 meters in a way to try to evaluate the efficiency of this strategy in this setting. And I just want to highlight that most of the RDT negative PCR positive individuals are within the index case house. Very few people are identified, both actually by RDT and by PCR, beyond the index case house. There are some, but you can see you had to screen 951 individuals to capture 17 RDT positives within the 140 meters. So based on this, I'm summarizing a lot of data very quickly, but we actually think a focal drug administration within the index case household would be a more efficient strategy, and have been communicating with the National Malaria Control Program about this. Now, we've used a 24 SNP barcode assay that was developed by Sarah Volkman and Rachel Daniels at the Broad Institute to try to do some genotyping of parasites. And this is some preliminary work that Julia Pringles led. And basically what I just want to highlight with this figure is that we are seeing similar parasite genotypes with this approach in the index case households that suggests that those index case households are related as transmission events, whether they were all imported together or whether there was local transmission we're working out. I didn't mention on the prior slide, but we do see higher numbers of Anopheline mosquitoes in these index case households. We're now working to try to genotype the parasites that are identified beyond the index case household. Our current hypothesis is that those beyond 140 meters are unrelated to the parasites in the index case household. Kelly Searles led a study using the same 24 SNP barcode to compare parasite genotypes from asymptomatic individuals that we find through active case detection with symptomatic individuals presenting at the health centers. And I just want to highlight a couple stories here very briefly. The blue dots here on this phylogenetic tree, if you will, represent those cases, asymptomatic individuals we think representing this chronically infected reservoir, and the red represents symptomatic individuals presenting to health centers. And by and large, these are two distinct populations. There's some overlap, but we think this perhaps suggests that these chronically infected individuals are not contributing to ongoing transmission in the area. And looking at parasite diversity in these two groups, we've seen a loss in parasite diversity in the asymptomatic, what I'm calling the chronically infected reservoir, over time, as if these parasites are being cleared out of this reservoir, but high diversity in the passively detected cases, we think this represents, at least in part, parasite importation. Now, to get a better handle on the issue of parasite importation in this area, Kelly Searle led a study where we gave a rather small number of individuals a GPS logger to carry for a month to track their movement. And what we saw on a broad scale, so these are individuals carrying a GPS logger for a month, tracking movement. What we saw was during the rainy season, the movement, the human mobility was rather restricted, and in the dry season, people were going on kind of longer journeys outside the district into other areas where there's higher transmission. We think that hypothesizing why the movement's restricted in the rainy season, this may be because people are at their farms, attending their farms, or bridges are washed out. But this may work to the advantage of malaria elimination because during the peak transmission period, people tend to have less long-term population movement. Kelly then overlaid the movement tracks on a risk map that we had generated earlier, so we could tell if people are moving in and out of high-risk areas. And the third dimension here is the time spent in a particular location. And we can actually look at where people are spending their time during peak biting hours in this setting. Lastly, I just want to briefly talk about documenting elimination, because that's a huge challenge in these areas with low transmission, and we've been using serologic approaches to look at this question. First, we use antibodies, measured IgG antibodies to whole parasite lysate, just a mush of the parasite. Tamaki Kobayashi led this work. These are serologic surveys conducted from 2000 to 2012, and you can see a slight right shift, a shift in older age groups over time, and we have plans to repeat this survey this year to see if there's been a further shift. Using a reverse catalytic model that Chris Drakely and his team developed, Tamaki and Isabel Rodriguez were able to show, using serologic data, a signature in serologic data, a decrease in transmission around 2007, 2008, which corresponds to what we've seen in the parasite prevalence data. And then finally, this was one of the ICEMR supplements where we collaborated. A number of ICEMRs worked with Phil Felgner and Hugh Davies using a protein microarray that detects antibodies to 500 Plasmodium falciparum parasites. This is ongoing. The analysis here is ongoing, but I just want to show you some broad patterns. This is our low transmission setting, medium transmission setting, high transmission setting. You can clearly see different signatures in the serology here. These are organized by age, with increasing age, young age to older age, and you can also see an age signature in the serologic data. And we're looking now at the specific antigens that are represented here and seeing if we can see different kinetics and dynamics in the antibody patterns. I'm going to move briefly. I know I'm speaking quickly. I want to cover a lot of material. I'm going to move to the high transmission setting. This is Nchelange District. And up till now, the National Malaria Control Program and partners such as the Bill and Melinda Gates Foundation have really focused on eliminating malaria in southern Zambia. And in fact, Zambia has set a goal of nationwide elimination by 2020. One of the real challenges to that area is going to be in northern Zambia. And our team has been evaluating the impact of various interventions there and trying to understand the epidemiology and vector bionomics that make control so difficult. First, our team, led by Maureen Kutsia from University of Witwatersrand, identified high levels of pyrethroid and carbamate resistance in the dominant vector Anopheles funestus there, also in Anopheles gambiae. I'll talk a little bit about that. On the basis of those findings, the National Malaria Control Center switched from a pyrethroid-based insecticide to an organophosphate, pyrimidine methyl, for IRS. And what they decided to do, because of limited resources, they decided to target the IRS along this lake, densely populated area along the lakeside, and based on health centers, the health centers are all located along the major road there. Based on health center data, population density, they decided to target the IRS here in this yellow area. And what I want to explain very quickly to you is why we think this is and will be an ineffective strategy. Marisa Hast has led this evaluation. This is our time series through active case detection in this area. Parasite prevalence on the y-axis, time on the x-axis. These gray bars represent rainfall data that we've collected. Very seasonal rain pattern, but there's transmission throughout the year here. Sprayed areas in red, unsprayed areas in black. Now, these are not obviously matched in any way. They targeted the high-risk areas. But the bottom line is we've seen very little impact of 2 rounds of IRS. I should have said the IRS with Ictelic is shown in these green bars here. Very little impact. So I just want to briefly explain why we think this is the case. First, we've identified widespread malaria risk. This is a malaria risk map for the area with uncertainty estimates here. Dry season, rainy season. This work was led by Jesse Pinchoff with Frank Carrero and Tim Shields. You can see very little difference in risk, and I should just say this is derived from our active case detection and remotely sensed data, ecological data. Very little difference in risk even in the dry season, despite the seasonal rainfall. So we've got this widespread malaria risk, even though they're spraying just along the lake here. Marisa Hast took our GPS logger study and implemented it in Nchelange. So these are movement tracks in dry season, rainy season. And what I want to point out is that people are moving back and forth from the inland area to the lakeside within each month. So it's very easy for them to carry parasites kind of back and forth. And Marisa also kind of generated this 3D plot showing the time people spend. This is very early analysis, but in this high transmission area, we actually had incident cases as people were carrying the GPS logger, so we could actually try to identify spatial patterns and movement patterns associated with malaria risk. This is the distance from their home during peak biting times, and there's a different distribution in distance from home for the incident cases for people compared to people who did not acquire malaria as they were carrying the GPS logger. Now, another major feature driving the challenge of malaria control in this area is the vector bionomics. And I can't do justice to this in a few seconds, but I want to show you the temporal and spatial patterns and why this confounds or I should say challenges the indoor residual spray strategies here. So we've got two vectors. Funestus is the dominant vector, but there's also Anopheles gambiae, and we have these distinct spatial and temporal patterns. What I want to highlight here is so these are vector count risk maps, vector count spatial patterns of the vector. This work was led by Jennifer Stevenson and Jesse Pinchoff and Tim Shields and Frank Carrero. You can see during the dry season, Anopheles funestus kind of peaks, and it's peaking in the interior. That's where the vector's found. So if you spray just prior to the rainy season along the lake, you really miss this vector. So this is another way the epidemiology and the vector bionomics can help guide the intervention strategies. I'm getting near the end. I just want to point out that Tamaki Kobayashi has led some recent work, conducted a cross-sectional survey in January, February of this year in Kilwa and Kashobwe on the opposite side of Nchelange, and we're trying to see kind of whether there's potential for cross-border malaria transmission here. This is also an area with a very high parasite prevalence, this by RDT, and actually very little vector control strategies in contrast to what's been implemented in Nchelange. So ongoing work in this area. I just want to end up talking about Mutasa District very briefly. Here we're trying to understand resurgent malaria and how you prevent resurgence. Similar to the story in Nchelange, our team, led by Maureen Kutsia, identified Anopheles funestus as the major vector here with high levels of resistance to pyrethroids and carbamates. So in Zimbabwe, too, as a consequence of the Southern Africa ICEMR work, the National Malaria Control Program switched insecticides to the organophosphate Actelic. They had a very different spray strategy. They sprayed high-risk wards, so not just a narrow area. And this is work that was recently published, led by Mufaro Kanyangarara, again, kind of time series. And here, after Actelic spraying, we have seen a dramatic decrease in parasite prevalence. It didn't eliminate malaria, but we see a blunting of these seasonal peaks, and we are continuing to follow this. Now, the big challenge here is that this area is right on the border with Mozambique, where there's been very little malaria control in that area in eastern Mozambique. And so we're going forward very interested in understanding cross-border malaria transmission here. Mufara created a risk map using our active case detection and ecological variables to try to understand the risk. This is, again, dry season, rainy season, uncertainty maps. What I want to highlight here is this high-risk area along the border in eastern DRC, along the border with Mozambique. And we're trying to understand whether this is being driven by the ecology, it's just better breeding sites, or whether there's a lot of parasite and human movement across this border. We know, and we've begun counting, enumerating patients from Mozambique seeking care at the health centers along this border. So our goal is to try to tease apart what's driving this high risk in the eastern border. Southern Africa, ICEMR is a work of a very large group of people, and I'm just presenting on their behalf. And I do want to kind of give a shout out to our doctoral students over the years who've really done a lot of the analytical work and led this, as well as the laboratory work. And I'll stop there. Maybe I went too quickly.
It was a wonderful presentation. I see that
Oh, go ahead. Okay. In your low malaria transmission area, based on your graph, you showed that there is a very low transmission, I think in the year 2005. starting from 2003, 4, 5, I think there is a very low transmission. Then there was a spike in 2006. After that, you have kind of very low level existence of malaria. Can you relate the spike and the low level malaria with the vector transmission, your genotype, and the anti-malarial treatment?
Related to what? If you could just repeat that last part. Related to
anti-malarial treatment, vector
Yeah.
transmission, and your genotype.
Yeah, so I mean, it's very tricky, as you know, kind of assigning causes to this temporal variation in the either cases or parasite prevalence. There was an ACT, the stockout of drugs kind of prior to that, and maybe that stockout contributed to that little spike. Again, I'm cautious to assign a single cause to that type of variation. There are obviously changes in rainfall that can drive the vector. One of the big mysteries, I'll say, or unanswered question is why there's been this dramatic decline in southern province Zambia in transmission. And I think the interventions have played some role, but I don't think they're the whole story. We do know that Anopheles funestus was a major vector there some time ago, and now we're only seeing Anopheles arabiensis, so there was a dramatic change in the vector, and I think that's perhaps attributable to drought in the early 2000s. So I think my own personal opinion is that that was a big driver in the overall transmission. But explaining a single spike, I can't do that. There was an ACT stockout.
But based on the 24 SNP genotype, can you differentiate the low-level parasitemia and the spike or the previous ones?
Yeah, I don't think we can do that using parasite genotyping. Joe?
Bill, thanks so much for just a wonderful talk. You didn't talk too fast, actually. It was very clear. I wanted to ask you about that phylogenetic tree between the active and the active and the passive case surveillance, because multiplicities of infection can be so high, but you're only showing a single genotype, and when you have low parasitemia, which you're going to find with active surveillance. Yeah. I'm just wondering what's the next steps, because it was so black and white, it was surprising to me.
Yeah.
So what next?
Yeah, no, that's a great question, Joe. And I didn't raise the limitations of the 24 SNP barcode, and we've certainly run up against that in terms of its inability to really deal with multiplicity of infection. We did see a much higher multiplicity of infection in the asymptomatics, those detected through active case surveillance, than the passive. Now that could in part just be driven by a dominant, parasite strain in the symptomatics at the time. Moving forward, we've established a collaboration with Steve Meshnick and his group at UNC, and so we're going to be applying a variety of new genotyping tools. Steve's been kind of promoting his VAR2 CSA genotyping, but maybe even some deep sequencing as well to try to get at that. But we recognize that as a limitation of the method we've used.